Right, to this evening's business. Darren is a professor at the University College Dublin. He enjoys the outdoors, and he's quite close to the Wicklow Mountains, so he can get out there quite often. And he's done research on birds and still gets involved in breeding bird surveys. He is a molecular ecologist. I had to look that up. I mean, I think I know what ecology is, but I wasn't sure what a molecular ecologist does. Perhaps he'll enlighten us. But I think, yeah, I looked it up on Wiki, which is always a good thing to do. Molecular ecology uses molecular genetic data to answer ecological questions, which seems very good. And ecological questions related to biogeography, genomics, conservation genetics, and behavioral ecology. So in the past, he's used this approach to look at a number of environmental challenges, including restoring forests and protecting bird diversity in the tropics. Tonight, he's going to take us through the work that he's been doing as part of the Irish Apicultural Programme to protect bees and pollinators in Ireland. So with that, I'll hand over to Darren and ask him to talk us through that. Thank you very much.
Great, thanks very much for introducing me. I'll just share my screen now. Okay, you should be seeing the presentation now. You can shout if there's a problem. Brilliant, and great. Thanks very much for having me. It's a real pleasure to speak to you all. As Bob said, I'm a molecular ecologist. So I use DNA to answer various questions and problems in ecology and evolution. In the last 3 years, I've been working quite a lot with pollinators in that way. We're all aware of the sort of pollinator declines, both of managed honeybees and wild species that people are worried about. And at the moment, I'm working with the Irish Apicultural Programme to investigate multiple stressors in Irish honeybees. We're working with Dr. Julia Jones in University College Dublin on an EU-funded project. So I'm sure you're all aware there's increasing fears about high colony losses and declines in hives of honeybees worldwide, particularly where their pollination services are really important for agriculture. Losses in Ireland for honeybees are actually quite high. You often get about 30% over winter losses. And the problem with this is hives face compound stressors from a variety of chemical, biological, and environmental factors. You know, you've got weather, you've got pathogens, you've got pesticides. So it's quite hard to pin down sometimes the root causes of colony losses. So the overall aim of the Irish National Apicultural Programme has been to look at multiple different stress factors and how they affect honeybees, and as much as possible do that in real world settings. So as part of this project, we've been doing an all-island sampling of pesticide levels in honeybees, and we've also been screening for pathogens in honeybees and apiaries across Ireland. And then with a particular focus on our dear old friend, varroa destructor. But first today, I'm going to talk to you about the work we're doing on thermal stress in Irish honeybees. So I think we're all aware at this stage that there's plenty to worry about in terms of the climate. So the pollinators are likely to face more extreme temperatures going forwards. And thermal stress is going to affect key aspects of colony health. So obviously movement and foraging, which is absolutely vital for a colony to feed itself. And then particularly the maintenance of a stable internal temperature and humidity in the brood nest area of the colony. This is really important for bringing through the next generation of healthy workers. And a lot of experimental data from the likes of Sealy back in the day show that there's these tight ranges of temperature and humidity, which honeybees need to maintain their brood at in order to get healthy broods constantly cycling through. So our key question was whether temperature would have an effect on bee worker movement or on the maintenance of brood nest temperature or humidity inside the hive. We couldn't do this, of course, in the landscape because thankfully temperatures haven't gotten that extreme yet. But what we were able to do was take advantage of some incredible facilities that our collaborators in Teagasc have. They've over a kilometre of temperature controlled glass houses where we were able to carry out temperature gradient experiments with our experimental honeybee hives. We also have 80 hives down in Teagasc and Carlow, which we use for different research to look at different stress factors or feeding in bees. So we selected 16 colonies of really similar strengths and standardized them to ensure they all had the same amount of brood and resources available. And all these standardized hives were set up in a bay, each with foraging plants and syrup feeders and water, and left for a couple of days to acclimatize. We did 4 replicates of this, and each replicate had 4 hives in it. So in total, we had 16 colonies just to look at sort of inter-hive variability. And then after they were acclimatized to the glass house and they found the food and so on and so forth, each replicate was run through a week-long schedule of set temperatures. These are based on the projected range of temperatures which might be experienced in Ireland in the foreseeable future. So each replicate was run through a day of 7 degrees, 12 degrees, 17 degrees, 22 degrees, 27 degrees, and then 37 degrees Celsius. And then the order in which they experienced these was randomized, so they wouldn't be able to necessarily acclimatize. And then nighttime temperatures were 5 degrees lower than the daytime temperatures. So to measure the effects of these temperature gradients on the experimental colonies, we used a couple of really handy little bits of equipment. So a Spanish collaborator of ours, the beekeeper David Atari, has developed these really nice counters which you just fit over the entrance to the hive, and it counts every bee that goes in and out of that hive. So then you can see what overall in and out movement is for the colony. And it just uploads that to the cloud and you can download the data. And then we put in these little iButton recorders, which are great. They measure the temperature and humidity every 15 minutes in where they're placed. So we put one on each side of the central brood frame to get a good temperature reading of what's going on within the hive when they're experiencing this temperature gradient. So let's go have a look at the initial results. So what I'm showing you here is the results of the 4 replicates. The solid lines, the purple, green, blue, and red one, are the mean score. That's the average measurement. And then the paler lines behind them is just measurement error. So what we found was overall an extremely positive effect of temperature on the numbers of bees going in and out of the hive on bee movement. So what that means is as the temperatures go up, then bee movement goes up, and it's got this sort of curved relationship. So as it gets quite a bit hotter, then the bees move quite a bit more. And there's definitely there's some variance between the replicates. The later 2, the green and the purple one, didn't show quite as dramatic an effect, but that was still a positive relationship that when they got hotter, they moved more. But and it's kind of hidden by the fact that the first 2 replicates had a really, really strong relationship with temperature. So basically, it seemed that when temperatures increased, worker bees were making more and more trips to get water to cool down the hives, probably. And what the other thing we looked at, the brood conditions, which I was really interested in, as that will be, a strong determinant of what is a healthy hive going forward. We did see an impact of temperature, which I suppose isn't necessarily surprising, but bees are known for their ability to maintain a very stable internal brood conditions. But as the temperatures got higher, we can see that we did see differences. So the top graph here is the hive brood temperature, and you can see as glasshouse temperature went up, the hive brood temperature also went up. And then the bottom graph is hive brood humidity, and as the temperature went up, humidity went up. Now they're still within what you consider approximately the safe range for brood temperature and humidity, which Thomas Seeley would have said back in the day, approximately between 57 and 63% humidity, about between 33 and 36 degrees Celsius. However, we are seeing this relationship, so you can imagine extrapolating this further. If you get more extreme temperatures, you're starting to get into dangerous range. There's only a limited capacity maybe of the hive to cool when exposed to heat. So just to summarize our preliminary results with our experiments to look into the effect of thermal stress and temperature on honeybee hives, we found that the foraging trips increased with temperature, likely workers seeking more water. We found the brood temperature and humidity did go up with increased temperatures, though with the temperature range we looked at, it was still probably within the safe range for honeybee brood. So within the immediate future, we're talking the next 30, 40 years, maybe, with projected climate change and temperature increases, it's probably not an immediate worry. But if temperatures continue to climb, you could see how the thermal stress, even in our part of the world, would push honeybees quite a lot, potentially add to stress factors as they try and keep the brood temperature and humidity within safe values. So we think there's quite an interesting preliminary work on the temperature tolerances of honeybees, but there's definitely further work needed, probably looking at what are the effects of a greater temperature range. And also for this, we've just looked at the Irish subspecies. So other honeybee subspecies are likely to have different tolerances. So researchers in other regions could carry out similar experiments with their local bees. And we've started to feed in some of this experimental data into this Behave modeling software, which a collaborator of ours uses in UCD. Quite an interesting one. So it basically models real world impacts based on some input data. So he's looking at greater temperature ranges to see hypothetically how that could affect bees, but that's still pretty early days. Anyway, so that's the temperature. Now I'm going to move on to our national sampling to look at first the impacts of pathogens and then pesticides. So honeybee hives present an incredible resource base for any pathogens and pesticides. So they carry often quite a high pathogen load. There's massive resources there in terms of honey, brood, workers. It's really a pathogen's dream. Therefore, the impact of pathogens on honeybees has long been acknowledged as quite a serious threat. That goes back to sort of long-term endemic pathogens, which had a big impact on the beekeeping world, like historical Isle of Wight disease, which is still debates about to this day. And then more recent emerging threats and sort of recently globally established pathogens like Varroa. And the big problem is that co-infection by these multiple impactful pathogens can have this multiplicative effect on honeybee hives altogether. And you guys probably are aware of a lot of this, but the heavy pathogen load of managed honeybees is in part due to the success, really, the movements of managed hives from their native range in Africa and Western Eurasia to East Asia and the Americas and elsewhere. Once us humans realized how useful honeybees were, they've been moved worldwide. The different strains have been traded extensively within the original range. And so that basically means that every natural pathogen they ever had anywhere has been spread everywhere. And then they've also picked up some very impactful pathogens from related species, which are very vulnerable to because they haven't that evolutionary history of dealing with that pathogen for a long time. In particular, they seem to be very vulnerable to pathogens picked up from the Asian honeybee, Apis cerana, as they're quite similar species. So the pathogens seem to be able to make that jump But until we moved honeybees into the Asian honeybee range, those pathogens never had the chance to affect the Western honeybees that we use for beekeeping. So this has led to a situation where you've got several impactful honeybee pathogens distributed almost globally. Most infamous of them, of course, is Varroa destructor. It's jumped over from the Asian honeybee and has wiped out pretty much all wild and feral honeybee colonies in a lot of Europe. Still actually get some feral/wild honeybees in Ireland, but it's not that common in a lot of the range of the honeybee. And a lot of honeybee hives only survive due to intensive management of Varroa, despite potentially there being some maybe resistant strains emerging. And of course, you have the tracheal mite, an old pathogen of honeybees associated with the historical Isle of Wight disease. You've got the gut fungi, Apis, Nosema apis and Serranae, which can cause gut lesions and dysentery in bees. Got the two gut trypanosemids, Critidia melliferaceae and the Maria passum, which can colonize and block up a honeybee's intestinal tract. You've got the bacteria which cause American and European foul broods, which turn your brood into sticky goo and usually require the hives, unfortunately, to be burned in Europe. You've got chalk brood fungus, which can cause bee brood to degenerate into dried out mummies like the ones shown. You've got a variety of Iflavirus and Dicostravidae viruses, which can cause paralysis and loss of function in bees. So clearly you've got a complex selection of pathogens acting on bee health and welfare, often together. So our aims are to understand how these pathogens might impact bee health individually and cumulatively. So our first objective was to map the distribution of all common pathogens across the island of Ireland. Only a couple of the more easily identified pathogens were well mapped in Ireland. Most weren't particularly well known. And then the ultimate aim was to understand the impact of these pathogens on honeybee colonies and what combinations of pathogens contributed to colony loss. And to achieve these aims, we coordinated a network of beekeepers to collect samples from apiaries all around the island of Ireland. So each beekeeper contributed samples of worker bees, caproot comb, pollen comb, and honey from up to 5 hives in their apiaries, or all hives if the apiary were less than 5. This allowed us to screen for pathogens in real-world settings. Our sampling network included 159 beekeepers, covering all regions and habitat types all across the island of Ireland. Subsection sampled their hives in 2020, and then in 2021, all apiaries in the study were sampled. And doing this sort of work in Ireland is, as well as kind of having this applied research element for beekeeping, it's really interesting to look at the evolution of pathogens and honeybees together, because it's a semi-closed system. Obviously, people still carry bees in and out, even though you're not supposed to so much anymore. But Ireland probably has much less transport in and out of new pathogens. So you can kind of see how these relationships build up over time. But yeah, anyway, we couldn't have done this work without 159 fantastic volunteer beekeepers who did most of the hard work for us, really. So the process was we sent out standard sample kits and equipment to all our participants who coordinated sampling locally. Then we arranged for cold train supply back to UCD for storage at -80, which was a really key step to ensure the preservation of these RNA viruses for later detection in the lab. The RNA viruses they're incredibly robust when they're in a bee, but the minute the bee dies, they tend to fall apart. So it can be hard to screen for them effectively. So in this way, so we need them to be preserved really well. But yeah, this sampling network allowed us to sample like at a national scale, at an island scale, quite effectively. And all collected samples underwent an in-depth pathogen screen, including multiplex PCRs, which polymerase chain reaction. It's a molecular method to identify different species or different viruses from their DNA or RNA. And we use this to screen for all of the pathogens I mentioned earlier, including all the common mites, bacteria, fungi, viruses. And additionally, we did detailed Varroa mite counts to quantify the number of Varroa per worker bee. This kind of allowed us to get a really nice detailed overall picture of the pathogen community across Ireland. So the first step of our screening was to map where these pathogens were, how well distributed they were, what were the pathogen dynamics in the modern Irish landscape. And here I'm showing you some maps where the large red circles represent an apiary where the pathogen was detected, and the small bluer circles represent one where it was absent. They're pretty big circles, as you can see, and they're a little offset from center. So basically this is, you can't identify where these apiaries were as part of our research. But the circles are just a general indicator of regional spread. They wouldn't be taken as exact locations. So as expected, what we found was that Varroa destructor was extremely common. You get that in about a third of apiaries around Ireland. Interestingly, in contrast to tracheal mite is almost gone. It was thought to have a really major impact early in the last century in Ireland, in the 1920s or so. There's big hive losses back then, and a lot of Dutch bees were imported to replace hives which are lost, but these days it seems to be quite rare, relatively speaking. And we found an interesting pattern in the Nosema fungal pathogens as well. Nosema apis, probably the more traditional Nosema pathogen, which has affected bees for over a century, was relatively uncommon in Irish apiaries, maybe 10%, which really contrasted with Nosema ceranae, which was more than 40% apiaries. That's a relatively recent import from the Asian honeybee, and it was much more widespread and common. It's pretty interesting. It could be showing that honeybees don't have that built-up resistance to Nosema ceranae. And in a similar vein, the traditional honeybee gut trypanosomid, Curtidia mellificae, was found to be completely absent from Ireland. We didn't get a single case. Well, Lotmaria passum, which is trypanosomid, which was only identified as a species in 2015, was found to be incredibly abundant. This was the first time Lotmaria passum has been identified in Ireland, and it's actually the most common honeybee pathogen on the island, being found in over 60% of apiaries. It may have avoided previous detection because it's quite hard to pick up unless you're using PCR. It's pretty hard to pick up even on a high quality microscope. It's also possible that Lotmaria is benefiting from the spread of Nosema ceranae, as it's been implicated that has a kind of a relationship there, that Nosema ceranae breaks the gut wall in certain ways that Lotmaria can make use of. So maybe the two are helping each other out. Some good news was that the Dicastrovide viruses were relatively rare. We had one case of Israeli acute paralysis virus, and we had now cases detected in this screen for acute bee paralysis virus, chronic bee paralysis virus, or Kashmir bee virus. So not all bad news. And then for the iflavirus viruses, Black Queen's cell virus and sackbrood weren't too common. Got a bit of sackbrood, all right. The most common one was deformed wing virus, which is pretty common across Ireland, and it cripples worker bees, as you can see from the image here. In a lot of cases, if you've got a high load of deformed wing virus. What was interesting was that we didn't find the sort of tight relationship we thought we would between that and Varroa. Varroa is thought to spread deformed wing virus, but actually in a lot of cases you found them in different apiaries. Now that might be because the virus will hang on even after Varroa is knocked out by treatment. So it could have been originally introduced by Varroa treatment. And also, of the beekeepers we found deformed wing virus, not that many of them reported visible deformed wing virus. So I'd say it's quite common at a landscape level at low levels, but it's only when it really builds up that you get a lot of chronic ill health in the hive. And then unexpected, sort of no surprise, chalkroot was extremely common. It loves a wet, moist environment like Ireland, so it's almost ubiquitous in Irish hives. Particularly notable was the unexpectedly high rate of foulbrood we got, actually. there was 9% of hives were positive for American foulbrood and 40% were positive for European foulbrood. This, of course, is potentially a big deal because it's a notifiable disease across the EU, which must be reported to relevant Department of Agriculture's, which often results in the hives being burned. But it's only notifiable when it's clinically identifiable. So if you can identify it under the microscope or you can visually identify it, then the hives have to be burned. The interesting thing was a few of these hives have subsequently been tested under high quality microscopes, and you don't see prevalent foulbrood infection. So what I think we're finding is basically the spores of both foulbroods are super common in the environment probably, but there's not that many chronic infections. So what probably happens is weak hives are taken over by these spores, which are hanging around in the background probably a lot of the time in a lot of the country, which is an interesting finding, which we'll have to think about more in terms of how we manage foulbroods. There's probably more prevalent than we thought it was, but probably usually not necessarily damaging to strong hives. So this is one we'll have to monitor closely going forwards. So what does that actually mean for hives? We found lots of pathogens, but what impact are they having in a real world setting? So to assess the potential impact of the pathogens detected, we asked all participating beekeepers to fill in and return COLOS forms for the years covered by the study periods. And the COLOS forms, as probably a lot of you are aware, collect data on colony loss impacts in a standardized way. This allowed us to model the proportion of hives surviving over winter against pathogen prevalence in that apiary and then other relevant environmental factors. The interesting result we found was 11 of the 12 honeybee pathogens we found weren't related to any impacts on colony survival. It didn't seem to have a noticeable overwinter survival impact. Of course, the one that did was our dear old friend Varroa. It had a significant effect on overwinter survival, and that effect also seems to be cumulative. So the higher the Varroa load, the lower the survival rate in that apiary. And if counts of mites per gram of water of workers. So this re-emphasizes the effect of Varroa. Really, it's been in Ireland since the 1990s, but you're still seeing this heavy impact on colony survival, even though a number of Irish breeders are hoping they've got fairly resistant strains nowadays. There still seems to be this general impact of Varroa on colony survival over any other pathogen. There are, of course, a number of caveats to these sort of results. It's always complicated to get standardized data when you're dealing with 159 individuals managing their hives in different ways. And there's certain stuff like we didn't find an impact of foulbrood on colony survival, which might be because if someone noticed a clinically identifiable foulbrood infection in a hive, they probably burned it immediately. So that wouldn't necessarily show up in colony loss data that we could collect. But anyway, what we did find was that these sort of subclinical foulbrood infections, which are probably more widespread, but maybe not so damaging, they didn't have a detectable influence on colony survival. And then the only other impact on colony survival came from the number of hives the beekeeper kept. Here again, we've got colony survival on the y-axis, and we've got number of hives kept on the x-axis here, the bottom one. And beekeepers who kept more hives had slightly higher overwinter colony losses. This probably just reflects that a lot of Irish beekeepers keep quite a small number of hives, less than 5, and they probably do it kind of more out of love than anything, and will really try and nurse through hives over winter. Whereas more commercial beekeepers who might have 100 hives, maybe they mightn't put as much time into nursing individual weak hives. It's kind of hard to tell always with these sort of things. So our overall take-homes for the pathogen work. We found many pathogens were much more prevalent than had previously been anticipated in the Irish landscape. most of these infections were probably not at a sort of a damaging clinical level. Most of them are probably kind of just background infections and are not damaging hives. But this illustrated that a lot of potentially damaging pathogens are very prevalent in the environment and probably will exploit weak hives. Some previously really common pathogens like Nosema apis are being increasingly replaced with equivalents like Nosema ceranae, which have jumped over from the Asian honeybee recently, which is interesting sort of like you see these disease dynamics. And then managed honeybees are less likely to have sort of built up resistance to these newer pathogens. So it's definitely one to watch. But on the main, we found honeybees seem to be quite robust to most of the pathogens. We didn't find these strong relationships to colony loss for most of the pathogens we identified, even with some caveats. Though what we did find is that varroa structure is still clearly quite impactful. When you have higher varroa levels, you have lower hive survival over winter. There's a very strong relationship. So it underscores that we still need to constantly be thinking about this guy and how to manage it, though I suppose it's something that all beekeepers do anyway. So the next step of this work is to integrate these results with the national pesticide screen, which I'm just going to talk about now, and see whether there's any relationships between these pathogens, and maybe some different pesticides might ease a path for certain pathogens. And then following on for that, our next plan is to look at the microbiota, so basically the gut microbes of the bees we've sampled, and see how these might be affected by different pathogens and different pesticides. So there's a lot of work that show that some honeybees can survive with a pathogen load, but it affects their ability to digest sugars and overall weakens a hive long term. But anyway, that's work for the future. So to the last work package I'll talk about, so one we haven't completely finished yet, but I'll give you some highlights, is the pesticide work that we're doing as part of the same national sampling as the pathogen work. So the same apiaries, we screened fully for pesticides and pathogens. So honeybees are exposed to a lot of pesticides in the environment from a number of different sources. You've got direct application of acarides and fungicides to hives at times to deal with different pathogens, particularly different sort of acarides to deal with varroa mites. So acarides are typically pesticides targeted at mites or spiders. And then, of course, probably the main route for more damaging pesticides would be herbicides or fungicides or insecticides that they pick up in the environment. If they're housed near apple orchards or the like, they can potentially be exposed to that. And certain pesticides, like neonicotinoids, for instance, are famously impactful on and lethal to bees. Hence why a lot of these neonics have now been banned, thankfully. And then, so a lot of the very obviously very damaging pesticides have been banned in the EU anyway, and in most of Europe. But now the complicated thing is there's a lot of pesticides which experimental data is showing it might impact bees in this way or that way if you combine them with different pesticides, but it's really hard to pin down landscape level effects and how they might undercut the strength of hives in concert. So most of them are still able to be used, and it's hard to know whether they're necessarily damaging to bees or not in a real world setting. So that was our plan to once again go back to these 159 apiaries and do a full pesticide screen and look at how these pesticides might be impacting bees in concert with pathogens and how that might affect colony survival and overwintering survival. So that meant back in the lab for an awful lot of processing samples, which was really interesting. By God, honey bee wax is something else. It's exceptionally good at breaking sensitive lab equipment. So an awful lot of this work was actually just manually processing out wax from pollen samples or from honey samples and then doing a lot of purification steps to extract it again in the lab and just clean up our samples to get pure pollen or pure honey to extract pesticide residues, which is what we did. So we screened for 360 different pesticides using both liquid and gas chromatography. We're really lucky that we got to use the really high-end Department of Agriculture food science labs, so we could blast through all these samples. The results are just come in now, so we're still processing them. But thankfully, we didn't get high levels of pesticides on the main in any of sort of at a national level. We detected 15 different pesticides, but the majority of them were only detected in 3% or less of the hives that we screened. So there's pesticides around, and some of them might be damaging, but on the main, the load of pesticides in Irish hives is relatively low. Probably reflects Irish agriculture. There's a huge focus on dairy and beef in Ireland, which has its own problems for bees, but it usually doesn't have the same sort of levels of pesticides spreading as other forms of agriculture might. The two common ones we did find were bosclate and propagate. So bosclate is a fungicide which can cause mortality in bees, but usually only at high levels. And all detections of bosclate we found in Ireland were pretty low levels, so by itself it shouldn't be too damaging. The bosclate you often get maybe on vegetables or sometimes in orchards sprayed, so potentially bees feeding in those regions might pick it up. You can see there's a lot of red dots around the Dublin region where we do a lot of our market gardening, so that's where the vegetables are actually grown in Ireland in the main. So maybe that's where they're picking them up. And then propagate is an insecticide, acaride, used on fruit trees or roses. We got a few detections of that spread around. It can be quite lethal to bees if you get high enough levels. We got some moderate levels of it. Usually it's only dangerous in combination. If you use it with other pesticides like dimentin or tricloran, then some bees can have high levels of mortality. But we didn't find those other pesticides in high levels. So by itself, propagate probably not too damaging, hopefully. But as I said, our final pesticide analyses are still to come. We've just gotten all the detections processed through, and we have to analyze it with the colony loss and the pathogen data to understand, is there any noticeable effect on colony survival of these pesticides, and is there any noticeable relationships between the pathogens and pesticides? There is some experimental work suggesting that certain pesticides can kind of open up zones for pathogens in infection by basically wiping out some of the bees' microbiota. Then external pathogens can get in, but we'll see what results we get from that. So watch this space. So maybe talking to you again. So just as an overall take home from the Irish Apicultural Programme research to date, we still have another year on this particular project. So we've got some nice preliminary results, and hopefully we'll be pushing out some more interesting research. We've highlighted that temperature impacts definitely do need to be monitored. Within the temperatures we're seeing within the next 30, 40 years, probably in this part of the world, honeybees will be fine, but they do seem to be definitely temperature affected. And if temperatures get more extreme, it could really start to stress bees, particularly when it comes to brood maintenance. Highlighted that Varroa is definitely still a significant threat, more so than any of the other pathogens we're finding. It's still having this heavy impact on hive overwintering survival, something that we definitely can't forget about. Not that I don't think beekeepers are in any danger of forgetting about it, unfortunately. What we did find, which was encouraging, was generally bees seem to be handling other pathogen loads relatively well. Even hives with a few pathogens on the main seem to be handling them well. It didn't have a heavy impact on the hive survival. And then pesticide impacts still need to be investigated in more detail. We've just finished our big screen, but we were pretty encouraged by the results. There doesn't seem to be massive prevalence of the more damaging pesticides, and we didn't get a lot of hives which had multiple heavy pesticide loads. So hopefully that we're not experiencing a very heavy impact in our part of the world anyway. So to finish up, I just want to thank everyone who funded this project, all our collaborators in Chagas, DAFM, IRAF in Spain, UCD, then all the Irish beekeeping organizations, Federation of Irish Beekeeper, Irish Beekeeper Association, Ulster Beekeepers, Native Irish Honeybee Society. All of them have had members who have contributed samples to this project, which we couldn't have done the work without. And I want to thank our excellent collaborators, particularly Danny Cho, who is our beekeeper who's absolutely key to all the experimental work. We've got Christina Botiaz, who helps a lot with pathogen work, Darren Stanley, and then Jim Garvey in Department of Agriculture, who gave us access to the chemistry labs, which really helped our work in that end. Okay, thanks very much. And that's with me, and I'm happy to answer any questions.
So thank you so much, Darren. That was fascinating. Yeah, as an analytical chemist, I was really impressed to see that you're analyzing for 360 pesticides.
Well, that's the equipment we got to use. To be honest, I can't take huge credit for that. I'm a DNA man who had to learn chemistry, basically, and thankfully Jim Garvey in Department of Agriculture was a fantastic mentor. But yeah, no, they've got some really, really nice kit in there, and we were lucky enough to get to use it for research.
Yeah. Right. So Darren's happy to take questions. That you could either raise a hand electronically or post something on the chat section. John Hill has already put something in. He said you detected EFB in samples from Northern Ireland and informed beekeepers, but the inspectors did not inform the bee inspectorate. Do you advise them to do so?
Yes, I have. I've also spoken to inspectors in the north, and I've told them the beekeepers are to inform them. So again, I think it's clinically identifiable versus identified by molecular means. There is a difference there, and I think Northern Ireland, they make that difference as well. But every beekeeper has been told to contact their local inspector to get it visually assessed. I think up there, I was speaking to, and I made sure to tell all the beekeepers they need to talk to them. So hopefully they have.
Yes. I spent 6 years as a seasonal bee inspector in Kent, and EFB, comes and goes, and so as you say, it's a matter. Very often, I think the bacterium is there, but not clinically visible, and not picked up by the lateral flow devices either. So, yeah. AFB did surprise me a little more, because my understanding was once AFB spores are around, it usually drives to the death of the colony. But quite a lot of it there. Other questions, please. Caroline McDonald. Yes, would you like to unmute and ask your question?
Hi everybody. Hi Darren, thanks very much. I really enjoyed that. I'm Caroline McDonald. I'm from Irish Beekeepers Association, and you got my samples at one stage, Darren. Now I was trying to organize my audio here, but those 4 different temperature settings that you organized down in Carlow.
Oh, yeah.
You spoke about that very, very early on. Yeah. So were all those hives at the same temperature, or did you put them, say you had 4 different samples? So were the 4 at different temperatures, or did you put them all up to 40 degrees?
So all 16 hives were experienced the same temperatures at some point, but basically we split them into 4 groups of 4.
Yes.
And the idea is to randomize the temperature schedule because if you set them all in the same temperature schedule, then the argument is, well, they've acclimatized to that schedule.
Yes, okay.
So they all experienced the same 7 different temperatures.
Yes, but randomly.
There was 4 different rounds of that, each with a random order.
Yeah, okay. So thanks very much. And thanks for the congratulations on your work.
Oh, thanks very much. And thanks for your help, Caroline.
Could I ask a question about the Lotmaria passim? We had a talk last year, I think it was, from someone who spoke to us at that time. I'd never heard of the thing before. You mentioned that it was very prevalent in the PCR testing, but that it was tricky to find microscopically. Do you know anything about how it, you can use microscopes? What in particular do you need to do? Is it the sample preparation?
So as far as I'm aware, if you've got a high-end scientific, like something like a scanning electron microscope or something like that, a properly high-end lab one, you can see it if you know what you're looking for. But like the sort of light microscope you can get in your own house, you'd be very hard pushed to find it.
Oh, okay.
Yeah.
Right.
But it's pretty new. I don't think it's well characterized in that way. There's only a few studies on it so far, but the ones which have tried to sequence its DNA to find it's gotten really common. But it does seem to be quite a recent thing. It might be this turnover from the traditional Critidia melliferaceae is declining, it's increasing, and that might be associated with that. Apis, the Nosema apis, Nosema ceranae cycling as well. Right.
The researcher who spoke to us was from Canada, and she said practically every place they looked, they found it. But I thought maybe in Europe we hadn't got to that stage, but maybe we have.
I'd say it's everywhere, to be honest. But it's kind of there's only sort of preliminary experimental work that does show it can be damaging to a hive, but I don't think anyone's showed that at kind of a real world landscape scale yet. But it's just not very well characterized. It's one to watch, I suppose you could say.
Yes. In a general way, how do you see these results providing benefit to beekeepers in keeping healthier colonies of bees?
Well, I suppose the results to the beekeepers themselves is probably for their own interests, and then maybe if they might want to cycle out hives if they've got a high pathogen load, replace the stock they have at the moment. I think probably, hopefully our main impact going forward would be how we think about how we manage the pathogens in Ireland and elsewhere. I think what would be a good idea is to set up a much more regular, intensive screen for foul breeds in particular, because I think we're only getting a proper handle on how prevalent something like that is now, and that will help us manage it. So I think it's more medium to long-term impact for beekeepers, showing how much is there if you really look closely, and then also how we think about managing what is a clinical infection versus a subclinical infection. And there's way more subclinical infections than we would have thought, and how we think about managing those going forwards. So it's kind of an emerging picture, I think, but I think it's all about long-term monitoring of these things, because we get kind of snapshots, I think, for a lot of these pathogens, which is only helpful to so far an extent.
Yeah. Okay, thank you. Neville Dearden has a question. Would you care to unmute Neville and put it to yourself?
Hello, good evening. Your slide of hive content being burned due to American foul brood or European foul brood showed the fire was on the surface of the ground, whereas we're told to burn in a deep hole. Is the whole fire method justified on grounds of effectiveness? Because I've never understood why we do that.
Well, yeah, I think I picked those pictures because they're more visible, to be honest. I'm not even sure if they were necessarily hives being burned for foul brood. I wouldn't be an expert on that. It does make sense to me to burn a hive in a hole, maybe. So, but I'd say, yeah, I'd probably defer to someone senior in a beekeeping organization who has more experience with burning hives than I would have, to be honest.
I think the hole is important because sometimes there's an awful lot of honey and it doesn't burn very well.
Right.
And therefore, you know, and it spreads all over the place if you don't dig a decent hole, and then you can bury it all quite nicely. So I think Neville, the hole is a good idea. Veronica. Are you there? I'll put your question then. You missed the name of the parasite mentioned. Oh no, it was Lotmaria passim you were talking about, wasn't it? Instead of, I can never remember that one, Cryphida mellificae. Yeah. Any more questions, anyone? Or should we let Darren go? Robert has a question.
Thank you very much for the talk, Darren. Before Varroa came to Europe, virtually every hive that we looked at would have had the symbiotic fly brawler seeker in it. Has that completely disappeared now from Ireland as a result of anti-Varroa treatments?
I don't think it's thought to be very prevalent. So when we looked at our list of pathogens which were worth prioritizing, we left it off. Basically, it didn't sound like it was common enough to check for. Potentially we should have included it to make sure it's absolutely gone, but yeah, it's not something you hear about very often. It probably is this Varroa being treated so aggressively, and even potentially like the seamers, maybe Varroa is taking some niche space there that the fly otherwise would have used.
Yeah. It always used to clean the bees' mouth parts and made it easier for them to keep feeding when they got a lot of pollen around the mouth parts, which locked up the mouth parts. And I took some film of this a long time ago, and you could even, once the brawler fly had cleaned the bee's mouth parts, the bee would actually exercise its mandibles after being treated, and then go back to its job. So I wonder what the effect has been for it to lose brawler.
Yeah, you get
Wow.
You get some interesting sort of carry-on effects for things like this. It can be hard to tell what these treatments and breeding will do. There's even suggestion that by breeding bees, I think there's a recent interesting paper that we've bred bees to use less prophylaxis, to kind of spread less antimicrobial stuff around their frames, because it makes for a cleaner frame and easier to handle. Our breeding has kind of resulted in that, but actually it's made them more vulnerable to Varroa. The ones which spread prophylaxis all over the place seem to be more resistant to Varroa. So sometimes you do have these carry-on effects of the breeding and treatment methods. I'm not sure about the fly. That's an interesting one to think about.
Yeah. Halfway between us and you, the Isle of Man, that's where you go if you want to see brawler.
Right.
Yeah. John Hill.
Well, I was actually going to say, as Roger Patterson was before me, actually, I'll defer my question till after Roger's spoken.
Go on then, Roger.
You're on mute, Roger.
On mute.
Right. Yeah, thanks, Darren. I've removed several hundred what I call free living colonies over about 50 years. And what I find is that they're much healthier than managed colonies. Now, my guess is that nature is quickly taking them out. So if there is any chalk brood or sac brood or anything like that, that is at a very, very low level. not like I see in managed colonies. And I'm wondering if beekeepers are doing the bees a disservice by what I'll call mollycoddling, management techniques and medications and that sort of thing, keeping bees sort of falsely alive that may not survive in the wild. Any views on that?
It's a really interesting one, and it's really hard to tell. You probably need to do like a giant 10-year experiment with hives and free living hives in like the same environment. There's guys in the southeast of Ireland who would say very similar to what you've said, and they never requeen either. They basically allow hives to live or die on their own feet, and they believe they have a varroa resistant strain now. It's really hard to prove that without quite a big experiment. It's entirely possible. That's natural selection in action, of course, isn't it? That if you allow the population to be impacted by a new stressor, and the ones that survive are the only ones you breed from. But it's just sometimes that doesn't work, and the population just dies out if the selection is severe enough. Yeah, it's really interesting. And I think it's maybe somewhere we should be looking to go in the next 10 or 20 years. A big landscape scale experiment with 500 managed hives and 500 managed hives from reliable strain, and just which one does better in that time frame. So you're on mute now, Rob.
Mute, Bob.
Sorry, John.
Right, thank you very much indeed, Darren. It was a lovely talk, very good talk, I have to say, very interesting. One of the things you said, you're looking at 360 different potential pesticides or different agrochemicals, I should say. Is anybody looking towards the idea of synergy, where you get two chemicals come together, and rather than their effect being of a particular type, it's multiple effect rather than additive? And I wonder, is anybody going to start to look at that? The trouble is, I know, is that the combinations of permutations are astronomical when it comes to that.
Yeah, so that's what we're trying to get at to an extent. So that's why we've gone basically island scale to try and take in samples from as many apiaries as possible in different environments. So I didn't get into the weeds of the modeling with the pathogens, but that's what we've done as well. So we've modeled each individual pathogen and also every iteration of the pathogen interacting with another pathogen to see whether you have these multiplicative effects. And we'll do the same for the pesticides. For the pathogens, I didn't find multiplicative effects. It was pretty close between chalkbrood and varroa, that maybe there was a slight edge given by chalkbrood to varroa infection, but it wasn't significant. And then I'll do the same with the pesticides, but I'll have to admit I haven't done that analysis to see whether if this pesticide multiplied by this pesticide multiplied by this pesticide does have an effect on colony survival. Now, with our data, we won't pick up any subtle effects like, affecting their digestion or their cognition or their foraging necessarily, but we should get the broad effects on overwinter survival proportions.
Thank you.
Thanks, Darren. So can we take one more question? I think Jeremy Tyler has a question. Jeremy, could you unmute and put your question?
Sorry.
Perhaps not.
Okay, on the early slides, you had the, my mind's going back now, but basically you had the slide with the bee count with the 4 different things in the colors, and then the next slide you had the brood. It seems like the brood and the brood increase. The purple line, group 4, had the least bees going in and out over the period, even with the temperature. However, it had the highest increase and highest base level from the increase, from what I could see. Would the temperature have anything to do with that, maybe in the development of nurses to foragers so that there's more nurses in thing, which would then lead to a higher brood increase, if that makes sense.
Yeah, I get what you're saying. So my guess for that one is that it was maybe less vigorous colonies on the main, and they weren't as capable of making allowances for the temperature. So that though the purple replicate, all of those hives started off with the highest humidity and highest internal temperature anyway, and then they increased quite a bit, whereas the other hives stayed at in the other replicates, those hives stayed at a lower overall temperature and humidity. And my guess would be that the other hives compensated for temperature by worker activity, basically by bringing back more water and doing more fanning. And then I think the purple replicate had the weakest hives, and they weren't as capable of using their workers to make up for the temperature effects on the brood. The workers were relatively inactive in those hives in comparison to the other replicates. Yeah, it's my interpretation anyway.
Okay, right. Well, I think we'll draw it to a close there. Thank you once again, Darren. Fascinating stuff. So yeah, let's. I don't know.
Thanks very much for inviting me. It was really nice to talk to you all. Oh, and for your first questioner, I'm going to follow up with my Northern Irish samplers to make sure that you get in contact with the bee inspector. I did tell them originally, but I'll make sure to get back in touch with them in case any of them have not. But yeah, thanks for flagging that up. And so yeah, it's always useful to talk to more beekeepers because you find out things. But yeah, thanks for inviting me. It was really nice to give a talk.
Many thanks. So cheerio, everyone. I'll leave you with some of the upcoming events, and hope to see some of you at our next event, which is the National Honey Show. So, good night all.